Organs like this pancreas are complex structures made up of various tissue types. We know if we look further into the pancreas, it has cell types such as alpha cells and beta cells. The question we ask is, how do these cells make up a tissue and eventually construct the pancreas? Well, from the earliest stages of development through to the adult, groups of cells constantly change the behavior of adjacent cells, such as change in shape, mitotic rate, or fate. These cell behaviors include adhesion, migration, differentiation, and division, and are regulated by cell signaling. The close-range interaction between two or more cells or tissues that allows organs to be constructed is called induction. In this interaction, there are two components. These two components include the inducer, which is the tissue that produces the signal and changes the other tissue's cellular behavior. This signal is often a paracrine factor, which are proteins synthesized by one cell that diffuse over small distances to induce changes in neighboring cells. The second component is the responder. This is the tissue that is being induced. It is important to note that not all tissues may respond to the inducing signal. The ability to react to a particular inductive signal is known as competence. A signal released by an inducer is useless if there is no means of reception, so an inducer must have a responder. Cells of the responding tissue must have a receptor, such as a transmembrane protein, in order to receive the inducing signal, as well as competence, which is the ability to respond to the signal. So let's talk about this example of the optic vesicle in the model organism Xenopus. Eye formation is a very complex process, and we know that not all tissues can respond to the signal being produced by an inducer. First, in eye formation, there is a tissue that is necessary to cause the lens to start forming. That is the optic vesicle, which induces the lens, which acts as the responder. However, if you took the optic vesicle and transplanted it to the lower region of the embryo, such as where the trunk is at, the tissues do not have the receptors to respond to the inducing signal. In other words, they are not competent. If you remove the optic vesicle where it usually occurs in development, or if there is a newborn that is a mutant, such as one that does not produce the inductive signal, the tissue will not form as there is no inductive signal being produced. This means that the responder cannot do anything. The inducing tissue must be present for the responder to undergo cellular changes that leads to lens formation. If you take some tissues other than the optic vesicle and put it here, it shows that because it does not have the same inductive signal, induction will not occur. Some of the best studied cases of induction are those involving the interactions of epithelial cells with adjacent mesenchymal cells. These interactions are called epithelial-mesenchymal interactions. All organs consist of an epithelium and an associated mesenchyme, so epithelial-mesenchymal interactions are among the most important phenomena in nature. Once you are successful on the DAT and go off to dental school, you will learn that the concepts of induction are crucial components of tooth development, which is regulated by epithelial-mesenchymal signaling. Interactions between the ectoderm and underlying mesenchyme in the dental lamina regulate tooth morphogenesis. These interactions cause the oral ectoderm to thicken, bud, grow, and fold to form the complex shape of the tooth crown.